I'm excited to be here today to share some of the work that we do in the Maynes lab, which revolves around harnessing the regenerative potential of the pediatric heart to target metabolic dysregulation in heart disease. So metabolic dysregulation is one of the hallmarks of heart failure. Usually what happens in a healthy adult cardiomyocyte is that the majority of ATP comes from fatty acid oxidation, while the majority of the rest comes from glucose oxidation. But what happens in a failing heart is that this balance is shifted from fatty acid oxidation towards aerobic glycolysis, where now pyruvate is no longer being shuttled into the mitochondria, but rather lactate is being generated. And what I really want to draw your attention to is how the net ATP yield in the failing heart is so much lower compared to a healthy heart, which really highlights the idea of a failing heart being an engine out of fuel. Now, interestingly, a lot of these metabolic dysregulations precede structural remodeling in heart failure, which make them an interesting early target for therapy. So currently, there are no effective treatments for heart failure, but one hot area of research is looking at the beneficial effects of paracrine factors that are secreted from mesenchymal stromal cells and their effect in different models of heart disease. But the gap that exists currently in the field is what is the effect of these secretomes on cellular metabolism? And that's what we really wanted to focus on in our project. So what we did was that we collected secretome from pediatric cardiac explants, and then we used that secretome as a therapeutic agent in our models of in vitro and in vivo cardiac disease. And the reason why we picked pediatric population to collect our secretomes from is firstly because younger individuals have a higher abundance of these cardiac progenitor cells, and also that these secretomes that are collected from younger hearts have a higher reparative potential. Now, we have injected these secretomes into our in vivo model of ischemic heart disease, and we have shown that they can actually improve cardiac function and contractility. But for the purposes of this talk today, what I really want to focus on is how these secretomes modulate cellular metabolism. So we use two different models for our studies. On the left is our in vivo model, which is an LAD ligation. So after we ligate the coronary artery, we inject the secretome around the area of infarct, and then we follow these animals for 28 days before we sacrifice them and harvest the heart tissue. On the right is our in vitro model, where we use iPSC-derived cardiomyocytes, and we treat these cells with the secretome for 24 hours under minimal media and 1% oxygen conditions to mimic ischemia. And then what we did was we wanted to measure mitochondrial function in our in vivo model first. So specifically, we wanted to measure the integrated function of complexes 1 to 4 and 2 to 4 in the electron transport chain of the mitochondria. So here on the y-axis, the oxygen consumption rate is a measure for activity of the mitochondria. And in the two graphs, you're seeing the integrated function of complexes 1 to 4 and 2 to 4. And in both of them, what we see is that tissue that was collected from secretome-injected animals have a higher overall complex activity in these tissues, suggesting a better overall metabolic function. So then we wanted to validate these results in vitro. So we measured mitochondrial activity at baseline with cells that were treated with the secretome or cells that were treated with control media. And again, we observed a similar thing where cells that were treated with the secretome have a higher mitochondrial activity. And in line with an increase with a higher activity of the mitochondria, we also see a higher production of ATP in these cells. So overall suggesting that the secretome treatment can preserve mitochondrial function under ischemic stress, and we've shown this in both in vivo and in vitro models. Now, as one carbon source that feeds into the electron transport chain, we wanted to look at specifically pyruvate oxidation. And the reason why we're specifically interested in pyruvate is because of its relevance under ischemic conditions. So on the y-axis, the higher you are, basically on the y-axis, the more reliant the cells are on pyruvate oxidation. And we measured this both under basal and stress conditions. And what we see is that cells that are treated with the secretome have a higher reliance on pyruvate oxidation. So then to explain these results, we looked upstream of pyruvate transport into the mitochondria, specifically at intracellular glucose levels, and downstream at pyruvate dehydrogenase activity, which is important for conversion of pyruvate into acetyl-CoA. And what we saw was that both intracellular glucose and pyruvate dehydrogenase activity is actually increased in secretome-treated cells, suggesting that there is more than one mechanism in play when it comes to increased rates of pyruvate oxidation. So our working hypothesis is that under ischemic conditions, without secretome treatment, most of the pyruvate is being converted to lactate, and only minimally it's being shuttled into the mitochondria. But what happens when we treat the cells with the secretome is that now pyruvate is being shuttled into the mitochondria for oxidative phosphorylation, and hence we have a higher ATP production in the cells. And this is one mechanism by which we think that the secretomes are enhancing metabolic function in the cell. But what we're really interested in is which specific proteins in the secretome are actually responsible for this effect that we are seeing. So we have identified overrepresented proteins in the secretome through mass spec analysis, and we've shortlisted the proteins that we think are actually responsible for this beneficial phenotype, and we are in the process of putting these into encapsulating them into lipid nanoparticles and delivering them to the cell to see if we can recapitulate the same metabolic phenotype that we see with full secretome treatment, and hopefully we can use those as metabolic treatments for heart disease. With that, I would like to thank my supervisor, all the past and present members in the Maynes lab, all our funding sources, and everyone who gives me advice throughout this process. And thank you guys for listening. Thank you.